Okay, this is a short overview of our Center for Personified Microbial Therapy. Here you can see all the licenses we have got in order to certify the building and all the utilities for making personified microbiota. So when the individual customer comes, he can bring the sample of his fecal material to this young looking lady, Oksana, and this is the point from which the whole story starts. So she registers everything, and maybe you can see this is the window to pass the bacteriological material for biologists. From this point, there is no relations between the customer and biological sample. Okay, all biological samples are specially registered, computer marked, so we can follow all the materials using computer technology. Okay, now we can go to laboratory part of microbiome. Well, now I entered the laboratory part, and you can see the corridor and all the rooms where the analysis is taking place. As you might remember, the samples of our material go to the first room for bacteriological analysis of fecal material. Maybe you can recognize this window and Oksana sitting behind it. So bacteriological material goes to our microbiological workers, and here you can see that they can do safe work with the sample and can do the preliminary study of the bacteria, which can grow on different artificial media overnight, and we can first get them, put them on different agar plates with different selective media, and then grow in these thermostats at different temperatures. Well, on the next stage, we can go to the microbiological study room, where our bacteriologists can analyze individual clones which we grow. And we can study those clones on this special biosystem facilities, do microscopy. We can look at the petri dishes and store individual clones at the biobank. The material, after being grown, is isolated out of individual clones as chromosomal DNA, and this chromosomal DNA goes for the further analysis. We can move to second room for the analysis. Here is another room for microbiology analysis, and you can see several PCR machines, like machines for real-time PCR, for regular PCR studies, and PCR boxes for assembling PCR reactions. All the PCR data is analyzed by computer, and the gels out of regular PCR, when we can study the individual clones for the absence of the virulence factors, and we have special panel of DNA primers for the virulence genes of the potential probiotic strains, or autoprobiotic strain as we call them. The results of PCR is done in the next room. Okay, you can see two wonderful looking ladies working with DNA material on gel electrophoresis. And you can see that here you can see the gels and gel monitor systems, whether you can register the results and figure out which strains out of individual clones are good for making personal probiotics. After we decide that these very strains are safe regarding our genomic studies, they can be grown individually in special facility which is taking place in other room. We can go to other room for first bacteriological growth of the material. Well, this is microbiology 3. And you can see that девушку не снимаем. Okay, you can see some ladies working with the clones, and here you can grow the personal clones which have been selected as milk fermented food product, or can be lyophilized using this freeze drying system, and can be prepared as individual capsules for personal, like this or like this, for personal probiotic in the capsule form. So we have two different forms of personal probiotics which can be delivered to the customers, and then these forms can be provided them as personal probiotic in different cases of dysbiosis condition. Basically, this is it, but of course we need to have special functional rooms for preparation of the media, for utilization of all the products which need to be utilized, and a special room for the storage of the final product which can be delivered to the customers. We can have a short look into the rooms for preparation of the media. Well,
no. Внутри.
okay.
Внутри.
Okay. All right. Okay. So here you can see the room for making media and for cleaning all the dishes, doing media, autoclaving, the necessary browse for growing of personalized probiotics. And of course there is another room for utilization of the waste, which can be disposed later. So in short, that is the laboratory. But I think that this kind of a brief overview is not sufficient, but at least it can raise some questions which I'll be happy to answer during the personal conversations.